Hi dear students, today I'm just going to brief you on gene regulation in prokaryotes. This was previously taken as a seminar, but I would like to give my feedback on gene regulation in prokaryotes. Now, if you go to consider the genes, there are certain genes which will be constitutively expressed or which will be continuously expressed. And these genes are referred to as the housekeeping genes. And many of these genes are involved in the basic metabolic processes which are being needed by the organisms. So there are certain normal enzymes which will be needed for the basic metabolism of a particular organism. They are being coded by the constitutive genes. And these genes are continuously producing its end products or its metabolites throughout the life of the particular cell, irrespective of whether it is needed or not, because these are quite essential for the survival of the organism and these genes are being continuously expressed. But at the same time, there are certain other genes which need to be produced or which need to be expressed only when they are being needed. Now, for example, if you need to utilize a particular sugar called lactose, okay, in the case of bacteria, you can find that the enzyme needed to degrade lactose is produced only when lactose is being present. That is an example of a regulated gene. So you can have two type of genes, either genes which are being expressed continuously, they are called the constitutive genes, and as well as you can have regulated genes whose expression or the products of these genes are being produced only when they are needed. That genes are referred to as a regulated genes. Now these regulated genes can be of two types. They can be inducible genes as well as the repressible genes. Now if you go to look into the inducible and repressible genes, there are certain genes which will be induced by the presence of certain agents. Okay, they are referred to as inducible genes. Now, what does it means? They code for enzymes whose levels will rise in the presence of small effector molecules called the inducer. If a gene expression is being found to increase by the presence of a small effective molecule, such as the inducer, that is referred to as an inducible gene. Now, for example, we have as I mentioned earlier, in the case of beta galactosidase gene, what happens? This is induced by the presence of particular enzyme, a substrate called the lactose. When lactose is being present, the beta galactosidase gene is being found to be induced. That is an inducible gene. And usually it has been found that these inducible enzymes, of course, the enzymes coded by the inducible genes are referred to as the inducible enzymes. These inducible enzymes are required only when the substrate is available. And they are usually absent in the absence of the inducer. In the case of lactose, lactose serves as a inducer for the beta galactosidase gene. Now you have another set of genes called the repressible genes. What are the repressible genes? These are genes which code for enzymes involved in the biosynthetic pathway, and usually what happens, they are products by the presence of certain molecules. What happens? The genes will get repressed over here. Okay, that is, I'll give you an example that will be more clear for you. Now, for example, in the case of tryptophan synthesis, okay, when there is more amount of tryptophan within the body, the organism does not need to synthesize what? Tryptophan. And so the enzymes which have been involved in the tryptophan synthesis should be repressed or being what is, it's just the opposite of induction. Okay, it should be repressed in the presence of tryptophan. Repressed means the production or the formation of that mRNA should be reduced in the case of repressible genes when tryptophan has been present. So the presence of the effective molecule over here, okay, which is that the effective molecule which was here was tryptophan. Okay, tryptophan, the presence of that is being found to down-regulate the pathway or the genes which have been involved in it. And usually, the genes which code for enzymes involved in the biosynthetic pathway are the ones which are considered to be as a repressible genes. And generally, these repressible enzymes are necessary for synthesis and are present unless the end product of the pathway is available. So, if in the case of tryptophan synthesis, if there is more amount of tryptophan, what happens? The enzymes needed for tryptophan synthesis are being inhibited, or the genes which are responsible for tryptophan synthesis are being repressed. Okay, but if tryptophan has been found to be absent or there is no tryptophan in the environment around the organism, what happens? The genes which are being needed for the tryptophan synthesis are being activated, or they start synthesizing mRNA for the synthesis of tryptophan, and they will become active. So the presence of tryptophan, the molecule, will repress a gene in the case of a repressible gene. But here, in an inducible gene, the presence of the effector molecule lactose serves as an inducer here, and it induces the gene. Now, I would like to tell you some concepts of gene regulation over here. You should, when you consider prokaryotic gene regulation, you should always remember in your mind that it is not a all or nothing phenomenon. That is, now, for example, if you have like lactose is come, it is not like that time you on the switch and lactose utilization is happening properly. It is not that. Neither that, it's not like full stop, a true full stop, all or nothing phenomenon. But it is in fact a case at which the level of mRNA synthesis is either decreased significantly and or it is coming to a very low level when you turn off the what? The lac operon or the a particular gene synthetic mechanism is being oft means the it is being found to be down or the mRNA synthesis of that is being decreased. Now, if you're saying when you're saying it is a repression which has been happening, or if you're saying induction is happening, what happens? It means that the mRNA coding for that that particular protein or the mRNA which has been found from that particular gene, inducible gene, is being found to be produced in a larger amount when you turn on the gene regulation. So the concept of gene regulation does not mean that in bacteria that the gene is totally switched off or totally switched on. It means that the expression or the amount of mRNA which has been coded by that particular gene is either increased or decreased. And another part that we have to note is about the decision making process. Now, the bacteria itself will determine on depending upon its environment whether it should what happens on the particular gene or it should switch off the particular gene. Now, if there is large amount of easily usable substances. Now, for example, glucose is something which can be used as a easy carbon as well as an energy source. Then the bacteria will try to utilize glucose for its metabolism. But only when glucose has been depleted and various other forms like lactose and all that is available, only then it will induce the genes which are being used for the synthesis of that particular protein such as a lactose. Now this is a depiction of it. If you have, this is about the regulatory decisions. Now if you are having a substrate for a pathway is being present, okay, fine. Then I will take, if it is a preferred and a carbon source, then it will yes. Okay, and then there are certain other enzymes, that is the catabolic enzymes are also there as well as the biosynthetic enzymes are there. If the substrate for the pathway is being present, it will go yes, and it is if it is preferred, it will utilize the same. Okay, so depending upon whether it should decide, like if the easily available substrate is available over there, it will utilize that particular genes. And if that is not there, what happens? It will decide that no, okay, I have to shift on to some other pathway. So it's based upon this that the gene regulation is being carried out. And I hope so this is clear to you. We'll be continuing about the various other aspects of gene regulation in the coming session. Thank you for now.